Now that we've talked about nucleotides and kind of what those individual building blocks are, now we can talk about nucleic acids. So nucleic acids, either DNA or RNA, are going to be polymers of nucleotides. So these are going to be nucleotides that link together by linkages, and if we look at the specific linkage here, it's one we've actually looked at before. So right, this linkage here actually showed up back in the lipids chapter. And this linkage here that links together two nucleotides is going to be a phosphodiester. So a phosphodiester linkage is going to be the bond that holds together nucleotides, whether you're looking at DNA or RNA. But this is going to be the type of linkage that holds together the two or more nucleotides. So a polynucleotide, whether it's DNA or RNA, is going to contain of a backbone that contains alternating sugar and phosphate groups. And then the bases are going to be what really distinguishes one nucleic acid from another. So the order of those bases are going to be what's different. And those bases kind of protrude away from this backbone of the sugars and phosphates. So just like for proteins, how we had kind of a directionality to it. If you remember, for proteins, we said that one end of a protein was the N-terminus and the other end of a protein was the C-terminus. For nucleotides, there's a similar type of directionality, except for nucleotides, one end of it is going to be called the 5' end. So right here's the 5' end, and the other end is going to be the 3' end. And the difference between them is that the 5' end, you're always going to have a phosphate, and on the 3' end, you're always going to have an OH, or a hydroxyl group. All right? And then the order of those bases, you're always going to read from the 5' end to the 3' end. And basically, whenever you're talking about DNA or RNA, whenever you're talking about the sequence of it, you're just going to read off the sequence of bases. So if you had, say, 4 nucleotides in a row in a DNA, you would say that the DNA sequence would be like ACTG, or adenine or adenosine, cytosine, thymidine, and guanosine. I can't remember the order I said it, but you get the idea. So here is kind of a structure that shows it a little bit better. So if you look up top here, this is going to be your 5' phosphates. This is going to be like the beginning part of your molecule with this phosphate hanging out. So this is going to be your 5' end. And then you have this backbone that alternates between sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then at the end here, you're going to have this 3' OH, and that's going to be your end of the molecule. So 5' end up here, down to your 3' end this way. So you're always going to read it going down in this direction. Now, this particular nucleic acid we would read as CATG, and that's because of the identity of the bases that would be there. Right? So CATG would be an example of the name of a particular structure. So this one, by looking at it, I can look over here at the sugar and see that there's no OH drawn here on the 2' end. So I would know that this would be an example of a DNA molecule, for instance. Right? So a strand of a DNA molecule, since there's no oxygens there. All right, so DNA. So DNA is going to be one of the types of nucleic acids that we're going to look at. So it won't be the one we're going to look at first here. So DNA was initially proposed way back in 1953 by these guys, Watson and Crick. They actually won a Nobel Prize for their work discovering this. And I always like to mention someone else, because there was another person in their labs who was working on this project who really did most of the work. Her name was Rosalind Franklin. So Rosalind Franklin kind of did all the experiments, kind of showed all the data to the main investigators in the lab, who are Watson and Crick. But she was kind of fundamental in doing the experiments and interpreting the data, and really did the most important experiments in determining what DNA was. And she's often forgot about because she didn't get the Nobel Prize for anything. And kind of more sadly for Rosalind Franklin was the fact that a lot of these experiments were done back in the 1950s using radioactive chemicals, because that's how they could kind of track where different things were. So for instance, she used a whole lot of radioactive phosphates to label her DNA. Well, it wasn't known back at the time that well, but working with radioactivity isn't really safe and can cause you to get cancer. So she actually died from cancer from doing the work that led to other people winning the Nobel Prize. So a big shout out to Rosalind Franklin for all that she did in terms of helping science get to where it is today. DNA has been found through those experiments to be a right-handed double helix, which basically means that there's two polynucleotide strands. That's why it's called a double helix. So there's going to be two strands. They kind of wrap around each other. So they go in opposite directions. One goes from 5' to 3', and the other one goes from 3' to 5'. And like we talked about previously, the sugar phosphate groups kind of form the backbone, and that backbone is going to be on the outside of the helix with the bases on the inside. So the words kind of paint part of the picture, but it's hard to visualize. So let's actually look at a picture of what it looks like on the next slide. Here's the structure of DNA. On the left, you can see like a ball and stick model, and on the right is kind of a ribbon diagram, which I think is a little bit easier to see kind of what's happening. So if you look at these two strands, you have to imagine, so the one strand is going to be 5' up here, and then if we follow that strand down to the bottom, that's going to be a 3' down here. So in other words, you can track your 5' down, and it wraps around this way, and it wraps around this way, and eventually it's going to end up over here. Now the other one, they run opposite of each other. So this one's going to be 5' down here, and it's going to go up and up and up, and then it's going to end up kind of at 3', let's say over there on the other side. So the blue ribbon part is going to be the sugar phosphate backbone, and then the bases kind of project into the middle of the helix. So the bases project into the middle, and if you look at the ball and stick figure over here, you can see the blue is kind of where the bases are, and they're almost even hard to see because it's partially obstructed by the backbone. You can see them clearly kind of shown in this diagram. So the one thing you'll notice about DNA is that the bases are kind of shown linked to each other. So this is what we call base pairing. So A is always going to pair with T, and C is always going to pair with G. One thing you might notice about that, you're always going to have a purine pair with a pyrimidine, right? So in terms of fitting those structures in there, you're always going to have like the same number of rings, right? Remember, purines have 2 rings, pyrimidines have 1. So whenever you're kind of cramming them together in the middle of this helix, you're always going to have the same number of rings whenever you're forming these base pairs. So A pairs with T and C pairs with G. All right, and the way that they pair with each other are through hydrogen bonds. So our good old friends, the hydrogen bonds. If you look at this C over here, here's an N that has H's attached to it, right? Hydrogens that are bound to N's can hydrogen bond, and there's a hydrogen bond with an O. Here's an N that has a hydrogen attached to it. It can hydrogen bond with this N. Here's an N with a hydrogen attached, and it can hydrogen bond with the O. So GC base pairs form 3 hydrogen bonds, and AT base pairs form 2 hydrogen bonds. So DNA that has more G's and C's is slightly more stable than DNA that has A's and T's, just because of that extra hydrogen bonding. So again, you don't need to know the structures. You should know that they're held together by hydrogen bonds. All right, so as I mentioned, a purine is always going to hydrogen bond with a pyrimidine. So we refer to these as complementary base pairs. So A with T and C with G.